Welcome to session number 4. There will be 6 presentations. First 3 presentations about population genetics of dermatophytes. Then other 2 presentations which will follow will be about diversity of onigenales fungi from environmental data. And the last presentation will be about population genetics on genome data. So let me introduce you the first speaker, who is Andrea Peano. Andrea, can you hear me?
Yes, I hear you perfectly.
He would speak about population genetics of Microsporum canis.
So thank you. Good afternoon, everybody. Greetings from Italy. I'm delighted to participate in this Congress, though unfortunately not in person. The next slide, please. So my presentation focuses on Microsporum canis, which is a very important dermatophyte species in veterinary medicine. It is distributed worldwide, and the cat is recognized as the most important host. Next, please. So you can see here an overview of clinical presentations in cats. They can be very variable, and notably, cats can present often with scarce evidence of lesions, which makes the cats very effective vehicle of infection for other animals and humans. The next, please. And of course, also dogs can be affected, and you can see here also different clinical presentations. The next, please. And of course, the importance of this pathogen is due to its high zoonotic potential. The next, please. So some years ago, we began to explore the possibility of straight typing of this pathogen using microsatellite markers. The next one, please. All of you know that these markers are widely used to straight type microorganisms because they are highly polymorphic. And since the panel of 8 microsatellite markers we have developed had very high performances, both in terms of reproducibility and resolving power, we thought that it was a useful tool to explore the dynamics of transmission. The next slide, please. For this reason, we began to build a database, including several fungal isolates from various countries and the hosts. The method, if someone is interested, the complete method is described in depth in the publication I mentioned in the slide before. And as regards the data analysis, I can tell you that each time we found a different multi-locus genotypes, which we call MS type, we assigned a serial number, a progressive serial number. The data were analyzed using a clone-corrected approach, which means that in case of samplings from cohabiting hosts, for example, in a kennel, in a cattery, or during an outbreak episode, and if we found, and nearly always we found the same genotypes for all the hosts, we consider just one for the calculation of the genotype frequency. We also tried to infer some population structure using some softwares, and we prepared a Google Map to allow an easier overview of the geography, of the distribution, the spatial distribution of the genotypes. Next one, please. These summarize the samples that are currently available in our database. You can see that we have many samples from Europe and fewer from other countries. And we have also samples from mainly 3 different hosts, I mean human, cats, and dogs. Next one, please. So out of this data set of strains, we found 91 different MS types, which corresponds to a great genotypic diversity, nearly 96%. And some of these MS types were found with higher prevalence, especially two MS types, the number 5 and number 90. And you can see summarized the most prevalent genotypes in this table. And please note that we also found a lot of genotypes which were found just in single occasions. If we go to the next slide, please, we can use the Google map to have an idea of the spatial distribution of these MS types. Unfortunately, I cannot navigate from here on this map, so I can just show you some screenshots. And I tell you that when the user accesses this Google map, he can decide, clicking on the box on the left, which genotypes you want to see. And if you go to the next slide, please, we can have the example, for example, of the two most prevalent MS types, number 5 in violet and number 90 in black. And this visualization allows to appreciate that MS type 5 is distributed mainly in Europe, from southern Europe up to northern Europe, while 90 dominates in Asia. The next one, please. The Google map is interesting also because it allows the user to see more data about the single samples. If you want to say more about one of the genotypes, one of the samples, you just have to click on it. And on the left, you can see information about the samples. In this case, for example, the isolate was collected in Firenze, in center of Italy, from a human being. Next one, please. Proceeding with other examples, we can see MS type 10, and from this visualization, you can see that it is distributed in Europe, but we have also found an isolate in Brazil. Next one, please. Just another examples. I don't have time, of course, to show all the genotypes, but we can see an interesting examples regarding the orange, the two genotypes. I don't see very well here the numbers, sorry, it's very slow, the picture. Anyway, these two genotypes are distributed, you see, mainly one in northern Germany and the other one in northern Italy. Next, please. Another option is also to tick all the boxes and to see a general overview of all of the genotypes. And this gives you another important information, namely that it's true that the MS type 5 is diffused all over Europe, but in Europe there are a lot of other genotypes. There is a large variability genetic in Europe. While in Asia, you see that the genotype 90 is nearly the only one present. Next one, please. This Google Maps is useful also because if you want, you can enlarge it and go to see very, very small particulars. For example, we have many samples for different metropolitan areas, and I show you just Turin in Italy and Paris in France, to see how even strains from very close locations, actually in the same city, there can be also a large variability, genetic variability. Next one, please. As regards the population analysis, I don't have time here to discuss in depth these aspects. There are different hypotheses, different models. I can just show you which could be now the most congruent findings between the different models of analysis. That would indicate that in our data set we have 5 distinct populations that you can see exemplified here with different colors. Next slide, please. We don't have now particular explanations to say why our data set is structured this way. Perhaps there are some underlying geographical factors that could be addressed just when we will have the availability of more strains from more remote countries. The next slide, please. A very important point is the possibility of a very important question that we are interested in when we make these types of genetic studies is to understand whether there are some lineages, some strains with higher zoonotic potential. And this is a good question also because in a past work, author hypothesized this thing, this feature, because they found that among human strains, among strains sampled from humans, there was a higher prevalence of virulent, of what they call the virulent genotype of Microsporum canis. If we go to our data on the next slide, please, we can see that you can see here the distribution according to host that we find on the different MS types in blue human, red cat, and green dogs. And you can see on the right something that may support this hypothesis, namely that there is a genotype more virulent, because you see that MS type 90 dominates the distribution sorry, I don't see. Can you hear me? Yes?
Yes, we can hear you.
Oh, okay. Sorry. That there is a genotype 90, which seems to dominate the human strains. But you must remind that this strain came from Asia, and from this continent, we did not have the availability of animal strains. So the overrepresentation of this MS type may depend just on a geographic feature and not on a real more pathogenic value for humans. On the opposite, the samples from Europe are much more balanced because remember that we have both humans, cats, and dogs, and they seem to tell another story because you see that on the left, on the graphic, that the blue bars that represent the human samples actually are more distributed over a lot of different genotypes, and that on the contrary, there is a genotype more represented, which is the MS type 5, but it is more represented in cats and not in humans. So these are these two possibilities that must be explored further with further samples. Next slide, please. So in conclusion, we can say that we have found some MS types more represented, and the hypothesis is that they may be more virulent, more adapted to a parasitic life, more resistant to drugs. These are all good hypotheses. We have found certain associations between some MS types and some geographical areas. We have found a great diversity between Europe and Asia as regards the number of genotypes. There are two hypotheses. The simplest one is that this is due just to the fact that in Europe we had much more samples, there was a greater intensity, and that if we will perform in the future the same intensity of samples in Asia, especially in animals, probably we could obtain more genotypes. The second one is that this is true. Actually, there are less genotypes in Asia than in Europe, and this may represent a more recent introduction of Microsporum canis in Asia compared with Europe. Some words also as regards the Google Maps. We think that this is a good tool for other researchers because they can assess it without problems. It is free, and they can have a look on this data, and eventually they could also in the future add their own data. And of course, this would represent a very great enrichment for appreciating the dynamics of transmission of Microsporum canis. Finally, we must say that we are continuing this analysis because as soon as we go on, we found new genotypes, and we understand that we need more samples to understand better and to address better all the questions that arose. And with the next slide, please, if you can, the next slide. I thank you for the attention.
One quick question. Are there some questions? So there are no questions, so, okay.
On the link.
That the diversity is related to European culture to keep cats and dogs as indoor pets?
Yes, this could be, of course, a reason. And yes, the reason is also that there is an increasing movement of animals throughout Europe, much more than in the past, and this could explain the veiculation of various genotypes all over Europe. And as I told before, all these hypotheses could be clarified when we could make new exams, and I like to examine also samples of animal origin from Asia and, for example, also from Africa would be very, very interesting. Thank you for the question.